Brain connectivity is a wide field. One can approach it in many different ways. First of all, the most general distinctions are between structural and anatomical connectivity, which we're not really dealing with here. So there's basically the hardware, the plumbing of the brain, usually with investigated with diffusion tensor imaging, non-invasively at least. Then there is functional connectivity, which looks at statistical dependencies of activation between brain regions based on correlation or spectral measures, phase lock and coherence, etc. So this is something, this is what I'm going to talk about now. But then there is also effective connectivity, where you are trying to infer causal interactions of activation between brain regions. That would be the domain of Granger causality and dynamic causal modeling. So this is not something I'm going to talk about in this session, maybe later. But even functional connectivity is already, there are lots of methods around, so there simply isn't the one thing that describes brain connectivity unless you really apply every possible method to your data. So you can look at directed and non-directed connectivity, and then there are different measures, correlation, mutual information. You can approach things, in a more sort of Gaussian framework or non-Gaussian framework. So, and then there are these different measures like coherence, phase locking value, phase locking index, more directed measures here like phase slope index and so on and so forth. So there is a lot you can do with your data, and I will only cover the basics here in this talk. This is a slide from Rezvan Farahiborg's PhD, just to illustrate how complicated it can be if you're really trying. So if you are trying to get some order into this whole zoo of different methods, there is no end to this tree. So you can always find different categories of functional connectivity methods, and then different subversions of different methods. So it is quite hard, but I think there are some methods at least that cover quite a range of these methods. That are now standard in the literature. So I will focus a little bit on coherence and the phase locking value. First of all, the first idea one may have is to just correlate time courses to see if there's a correlation between brain regions. So this would be, let's say, a naive correlation. So you may do this in a particular frequency band or not. Imagine you have your two regions here, two time courses. Why not just correlate them? Well, in that case, you'd be very susceptible, I think, to small changes in latency differences between brain regions, noise, different frequencies, that would, I'm not sure how interpretable this would be. I don't actually know if anyone has properly attempted that. Also, you wouldn't detect whether one signal is simply a shifted version of the other signal in time. So you might get zero correlation if this signal is just a little bit shifted with respect to that. So you might then have two orthogonal signals here. So this sort of naive correlation of time courses is usually not done with EEG and MEG data. It is something that I think in some cases is done with fMRI data, where you have a very low temporal resolution. So there are these matters of small latency changes, for example, wouldn't have a big effect. However, what people do, especially in the case of resting state analyses, is they compute the envelope of a signal. So you first need to bandpass filter your data into a certain frequency range. Then you compute this envelope. I had this in the previous session on, I think, the Fourier transform. I also covered that a little bit. If you do that, then you have sort of removed some of the high frequency bits from your data. If you do that from if you have that for two regions or even more regions, but let's start with pairwise relationships here, then you can correlate these envelopes with each other. So this is called the Hilbert envelope. And you can apply that. You can then correlate that between brain regions. Okay. Yes, and that is the basic idea behind most resting state analyses. So if you're interested in some of the aspects of resting state analysis, for example, look at the paper by Hilbert et al. It basically illustrates what I've just shown you. In this case, it also illustrates what is often done. You use a seed region somewhere, and then you look at the interaction of the seed region with voxels or vertices all over the brain. So you have one envelope for your seed region, you correlate it with all the other envelopes across the brain. This also deals with some issues with respect to leakage and spatial resolution, how that can affect functional connectivity. It's not something I'm going to talk about in this session, but it is an important issue. So we need to talk about that at some point. And it has produced promising results. So quite a seminal study here by Brooks et al. So they compared fMRI resting state networks. So you can maybe see here fcMRI with MEG networks in different frequency ranges. So this is alpha, for example, this is beta in the beta range. I think there's a beta range here. And then they find that some of these networks correspond. For example, here in the alpha range, the network looks similar to the one in the fMRI data. So there's sort of a frontoparietal network as a default mode network, and so on and so forth. So this has really proven to be a very useful approach to functional connectivity for resting state data. But then there's also another approach that is also maybe a bit more useful for event-related designs. So for these amplitude, for the envelope correlations, you need relatively long data segments. So it's suitable for resting state data, not necessarily for event-related data where you want to look at ideally millisecond by millisecond connectivity, but let's say time windows of 100 milliseconds or 200 milliseconds. So that is something you will have to decide also in advance, of course, what type of functional connectivity is suitable for you. But if you're interested in this kind of spectral connectivity in terms of synchronization, then there are lots of methods that are based on phase locking and coherence. I already had this study in one of my presentations before. So you may start with the assumption that different frequency ranges actually reflect different networks of the brain that may then also reflect different computations, different processes, and maybe they communicate via oscillations in different frequency ranges. So if that's how you approach your data, then you might be interested in the phase relationship between signals in different regions. So let's imagine you have two signals from two regions, red and blue are two brain regions. And if you can show that one of them is always a little bit behind or a little bit ahead of the signal in another region across trials, then you can conclude from that there's some sort of functional connectivity between these regions. So there's some stable phase relationship. So in the time domain, it would be a reliable or a consistent time difference across trial. In the frequency domain, there would be a consistent phase difference between signals. But the first assumption is that you are looking at that it's meaningful to look at your data in narrow frequency ranges. So this is only really meaningful if you look at your data in narrow frequency ranges like alpha, beta, gamma, and so on and so forth. And then we can look at our old friend, the polar representation of signals. So that sort of represents two signals here in the frequency domain. Remember, you can express periodic signals as this pointer going around the unit circle. And if you have two of these signals here, then you can, for every trial, compute the phase difference between these signals. And if you can express that also as a vector for every trial, and if that's consistent across trials, this vector always points in the similar direction, then you can say, well, there's some phase relationship, some functional connectivity. If this relationship is random, then you can say, well, there's no functional connectivity, and that's the basis for quite a range of methods. I'm not going to explain all of them, but the phase locking value is one of the most common ones, and this is illustrated in this nice tutorial here by Bastos and Schöfferlin, which I mentioned before. So imagine you have your different trials here. I'm sorry if this is a little bit small, this presentation, but you have maybe trial 1, trial 2, trial 3, and 4, and two signals from two regions. If they are all doing exactly the same in a particular frequency range, so in this case, two sine curves at every trial, then the cross-spectral density, which describes the relationship between these signals at every trial, will always point in the same direction. So these different arrows for the different trials always point, in this case, along the real axis of the polar representation. And if across trials they sum up perfectly, then you can say, well, there is a reliable phase difference across trials, and therefore there's significant functional connectivity. In this example here, these signals are not the same between regions, but one is always shifted in time, also shifted in phase, the frequency domain for every trial, and this shift this latency difference or this phase difference is constant across trials. So in this case, this relationship is represented by these vectors along the imaginary axis of the polar representation. But if you sum them up, you still have a significantly non-zero vector here. That also means there is a stable phase relationship across trials, and therefore there is significant functional connectivity. In the last example, if this relationship between the phase of these signals or the time difference is random across trials, this will be represented as vectors that if you sum them up, they cancel each other out, and you have no functional connectivity left. So that is the basis of most of these spectral connectivity methods. Just to highlight, there can be some confusion in literature. You can apply these phase locking measures to relationships between signals between regions, just like I explained. So this would be connectivity measure, but you can also compute this for one region, but across trials to see if a signal is always has the same phase relationship across trials. So this would not be connectivity. This would just show you that there is something phase locked to your stimulus presentation. So these are two different kinds of phase locking. It's just terminology at this point. So I'm obviously focusing on functional connectivity here. There's another method that is used a lot. So at this time, so far, I've only talked about phase differences, but what about amplitude differences? So the correlation I showed you for the resting state activity that looks at the amplitude correlation between signals. Now we focus on the phase, but you can actually do both. So what I just illustrated, what I just showed you there, these these vectors that you get for every trial that describe the relationship in the polar representation between two signals, between two regions here. So you can do that while taking into account the amplitude. For phase locking, you normalize all your signals so that all these vectors lie on a unit circle. So you ignore the amplitude, you're just interested in the phase differences, but the coherence takes the amplitude into account, otherwise uses a very similar logic. If you have these random relationships across trials, you get an almost zero coherence value. If you always have a relatively similar relationship between your signals across trials, you have significant coherence. In a way, and I think that's also what they say here in this tutorial, so coherence can actually be interpreted in a way as a amplitude-weighted phase locking value. So trials with low amplitudes are weighted down with respect to trials with high amplitudes, which you may or may not want. But at least in this case, with coherence, you test both. I mean, there's a bit of both amplitude relationships as well as phase relationships. So I think that if you just want to see if there's any kind of functional connectivity between two regions, then coherence is not a bad starting point, unless you have more specific questions, and there's a good reason why you want only the phase relationship. Maybe in a slightly down-to-earth way of describing what coherence does. So if one signal is a time-shifted and possibly rescaled version of another signal, then their coherence is exactly 1. So there's one extreme case. So imagine you have one signal in one region, and this is just a little bit delayed in another region, and maybe a bit amplified or a bit dampened. You would still have perfect coherence in that case. Of course, there will always be noise, but so this is at least what the coherence is testing, whether there's some relationship that doesn't depend on, unlike a correlation, which assumes that signals go up and down exactly at the same time. With coherence, you also take into account whether one signal can potentially be a time-shifted version of the other one. While if two signals are simply random and independent of each other, then their coherence is 0. So this is more like a more intuitive way of describing coherence. Just to illustrate the difference between phase locking and coherence. So I've just talked about coherence. This is what we do. We use these vectors in the polar representation. We include amplitude and phase and check whether it's different from 0. And for phase locking, we normalize these vectors. Apart from that, it's a similar logic. As you may already have seen in the previous examples, I mean, coherence or phase locking values were never really exactly 0. So it's quite unlikely that they sum up to exactly 0. And that is potentially a problem when you look at these signals, especially if you have conditions that differ in numbers of trials or for some other reason with respect to signal to noise ratio. So these values are always bigger than 0. So they're always positively biased. So even if you just have noise, if there's really no relationship between two regions, if you have only a limited number of samples, I mean, that's always the case. We never have infinitely many samples. Then even if you have a reasonable number of samples, and this is really just for illustration, this is not, this is still a very low number. So you will always get a non-zero sum of these vectors. And in this case, if you have very few trials, it means very unlikely that you pick 2 trials that sum up to exactly 0 or close to 0. So you will always get something that is biased towards positive values. So that means if you then test whether this is bigger than 0 or not, you will probably get a significant result in for many of these samples, for many of these selections of trials. That means if you have very few trials in your data in one condition, but you have maybe more trials in another condition, this may just look like you have high coherence or high phase locking just because you have few trials. So noise will have a bigger effect here than in the condition which has more trials, but this is just an effect of the signal to noise ratio. Okay, this is just described here. I mean, you can first do several things about that. So you can actually plot your connectivity metric for your baseline data to see if your effect is already, difference between functional connectivity measures is already present in the baseline. And if that's the case, then you may want to think about possible confounds like your signal to noise ratio. It's also common to equalize trials between conditions. So you pick basically you take the condition with more trials and you throw away some, which I think is a bit wasteful. So you are losing data, which is annoying. But yeah, in that case, at least you can say it's not the number of trials that causes any difference in connectivity, in your connectivity metric. You could also consider a baseline correction after you've computed your connectivity metric, compute coherence, in that case for the baseline interval and for the post-baseline interval or post-stimulus interval, and subtract the measure from the baseline. Has its own problems, but at least you are somewhat correcting for possible effect in the baseline. So you can, of course, first check whether there's a baseline effect. I mean, there's also some good news in the tutorial, Bassus and Schöfferlin. I mean, they looked at this kind of bias for the number of trials. I apologize that this is quite a small figure here. You might not see the plots here. But they show that this bias becomes relatively small for more than 50 trials. So make sure that you have a large number of trials and you might be okay. You can also plot something like this for your own data. So subsample your own data, maybe 10 trials, then 20, 30, and so on and so forth. Compute your connectivity metric and plot this, and you will see whether you are in a range that flattens here at higher number of trials, or whether you are somewhere in this range here where you may have to consider this as a confound. And there are some connectivity metrics that avoid this problem, which I am not going to talk about in this session. Also, nothing prevents you from computing these spectral connectivity measures for wavelets, for example. So if you're interested in sample-by-sample connectivity, well, compute wavelets. I mean, they basically give you an amplitude and a phase for every sample. When I introduced wavelets for the time frequency analysis part, we ignored the phase. We were only interested in the power. But wavelets produce both amplitude and phase. And when you are in business, so you can compute your polar, the relationships in the polar representation between regions. You can do that for all the different frequencies. You can do that for all your samples in your data. Of course, it takes longer, but you then actually get time-resolved functional connectivity. But with all the caveats that we mentioned before in the time frequency section, so you have a trade-off between temporal and frequency resolution. And you need certain numbers of cycles for your wavelets. So you have to make sure that they actually, that your latency ranges, your epochs are long enough. So you have to select your frequencies accordingly. But once you've done that, you can do time-resolved functional connectivity. And this can, for example, be used to compute either seed-based connectivity, we saw some examples earlier, or because it's relatively easy and quick to compute these. You can do something like an all-to-all connectivity, you know, take a parcellation of the cortex, lots of regions of interest here, compute connectivity, pairwise connectivity between all of them. So this could, for example, be an exploratory analysis that may then lead to further hypotheses that you may want to test with effective connectivity methods, Granger causality or DCM, or maybe this is actually a result in its own right. If I'm not completely mistaken, this might be the result for a visually evoked response. So that's why you see some strong connections here in the back of the brain, but you also see a lot of other connections here, which may actually be due to leakage. So spatial resolution, as usual, of course, is something you have to take into account when interpreting results from these analyses. Okay, so that was my bit about functional connectivity.